Hey guys, so we're officially on to Unit 6 now, which is called Meiosis and Karyotypes. The first thing in your notes is a little diagram. We're going to follow through and create a flowchart for that. So look in the section of your notes that says, how did we get here? This is that flowchart. So basically what's going on, we're going to start where it says puberty. So if you guys would put a little number 1 next to puberty. Basically, what we're talking about now, puberty is going to go through a process called meiosis. So I want you to label this arrow meiosis. If you don't know how to spell it, it is the title of your notes. Alright, so when we go through puberty as humans, we go through this process called meiosis. And meiosis is going to make something called gametes. Okay, you're going to spell gametes, G-A-M-E-T-E-S, gametes. Okay, so when we go through puberty, we go through a process called meiosis, which makes gametes, and we'll talk more about those here in a second. Okay, now gametes are going to get fertilized. Okay, so that's this blank right here. Gametes get fertilized during sexual reproduction. Okay, so sexual in this first blank, reproduction in the second one. Then sexual reproduction is going to create something called a zygote. So that's Z-Y-G-O-T-E, zygote. Okay, and we'll talk about that here in just a second as well. Okay, so sexual reproduction creates zygotes. Zygotes divide by mitosis, which is a process that we've talked about before. Okay, mitosis is going to make what we call an embryo. That's E-M-B-R-Y-O, embryo. Embryos become babies. Okay, so baby is this blank. And then it says babies then go through puberty. We start the process all over again. So puberty, the arrow we call meiosis. It makes gametes. Those get fertilized during sexual reproduction. Sexual reproduction creates a zygote. Zygotes divide by mitosis. Mitosis makes an embryo. Embryos become babies, who then start the process all over again. If you missed that, you may want to pause this video right here so you can copy down those things from this slide. Okay, so we've already talked about mitosis before. We're going to do a quick review of that, and then we're going to talk about meiosis, which is the topic of our unit this time around. So mitosis we talked about back when we talked about DNA and the cell cycle. If you remember, interphase, prophase, metaphase, anaphase, telophase. IPMAT with the hand signals, that was mitosis. Alright, so in mitosis, our cells are going to divide once. They go through that interphase, prophase, metaphase, anaphase, telophase once. Throughout the end of that process, we end up with 2 cells. We use mitosis as a form of asexual reproduction. So what that means is you only need 1 parent. You might want to write that down, hint, hint. We'll talk about these words here in just a second as we get further on in the notes. But what you need to know is that mitosis makes something called diploid cells. So with diploid cells, we're going to consider that 2N. And N, write this down, means number of chromosomes. So it's kind of like a math formula. We're going to have 2 times the number of chromosomes, which is what this bullet point says right here. Diploid cells are going to have 2 copies of chromosomes. Mitosis is used for growth and repair in our bodies. Very important to note that the cells here are identical. And quick review of this word. Mitosis creates somatic or body, B-O-D-Y, body cells. May want to write that down too. Okay, now meiosis, we'll talk about what meiosis is and why it's important here in just a second, but it's always good to have a comparison t-chart to come back to. Okay, so in meiosis, we're going to divide the cells twice. So we're going to have interphase, prophase, metaphase, anaphase, telophase 1, then prophase, metaphase, anaphase, telophase 2. So instead of just going through that cycle once, we're going to go through it 2 times. Instead of 2 cells, we're going to get 4. Meiosis is involved in sexual reproduction instead of diploid. Meiosis makes haploid cells, so it's considered 1N. So that N again stands for number of chromosomes. So this one only has one set or one copy of the chromosomes. Every cell that's made in meiosis is going to be genetically different, and it makes something called gametes. Now, again, I just told you at the very beginning, we will talk about what a gamete is here in just a second, so hang tight with me. Okay, so you've got a new bullet point in your notes that says, why is meiosis important? So number one, meiosis is important because without it, our species would die. Okay, and the second reason that it's important is that meiosis creates gametes. Now, another word for gamete is sex cells. There are two possible options to have sex of your sex cells, and we'll talk about those here in just a second. Okay, but basically without those gametes, okay, we would not be able to continue life. So two reasons is important. Without it, the species would die. And the second one, it creates gametes. So what the heck is a gamete? Basically, you've got another T-chart in your notes that compares somatic cells and gametes. Quick review of somatic cells. First off, we called those body cells. Examples, make sure you write these in the T-chart, would be your skin, your liver, your eyeballs, your brain, your heart, your muscles, your lungs. Any of those things are going to be body cells. Now, gametes are only sex cells. The only two that you can have are sperm, if you're a dude, and egg, if you're not a dude. Okay? Remember, we talked about this before. This is the DNA that you give to your kids. If you do not write that down, sorry about you if you do not pass this quiz. All right, so to talk about meiosis, we're going to spend a little bit of time talking about something called a chromosome. If you guys remember, chromosomes look like X's, and they're in the nucleus of our cells. Okay, so chromosomes are tightly wound DNA. They wind up to look like X's, and they're in the nucleus. Now, you have a picture that you drew in your journal. It looks a little bit like this. Okay, so make sure you label it. First off, the big button-looking thing in the middle, that's called a centromere, and its job is to hold the chromosome together. So this is kind of like an arm, and this is another arm. We'll talk about that in just a second. Okay, but those two things have to be held together by this button in the middle, okay, or else it would fall apart. The lines on the chromosomes, the bands, those are genes, so make sure you label that. And then at the bottom, you'll see a label that connects both of these in your journal. Basically, you want to label those sister chromatids, and say that those are the arms. And again, that's what the centromere is holding together. Okay, now the nucleus has the chromosomes, right? We've said it over and over again. So the nucleus of every different type of organism is going to have a different number of cells. Humans, we have 46 total. We got 23 of those from our mom and 23 of those from our dad. And they are arranged in pairs. So make sure you've got all of that written down under where it says total under your human chromosomes section. Now, those pairs are called homologous chromosomes. So definition here, homologous chromosome is a pair of chromosomes. They look the same, they're the same length, and they have the same genes. Okay, so look the same length, same genes. Okay, now each pair, if you'll look up here in this picture, is identical until there is a mutation. So I want you to go back to your yellow foldable that you glued into your journal in the last unit, and we're going to talk about those chromosomal mutations, okay, because I told you we'd talk about the second section of that foldable, okay, in this unit. So turn to that, and let's fill that out. Okay, the first one we're going to discuss is called a deletion. Draw this picture, A, B, C, D, E, F. Okay, in a deletion, all right, we're going to delete a gene. So like, then you'll draw an arrows to show that there's a change. This time around you'll have AC and then still DEF, but we deleted out the B, so that was a change. Okay, second type is called a duplication. So again, we're going to continue on with that same picture, ABC, DEF. Draw an arrow to represent the change. Now this time around in a duplication, we're going to duplicate or copy a gene. So we went from ABC over here to ABBC. Okay, so we added in a second copy of that B. Okay, next example is called an inversion. Now in an inversion, the chromosome gets twisted and switches genes. Okay, so you'll see this kind of looks like a pretzel. This kind of looks like a pretzel. It got twisted up, and it's going to switch genes over here. So instead of ABC, we now have AED, CBF. So again, make sure you draw that arrow to indicate change, and then the change in the chromosomes. Okay, now the weird one and the one that Star loves is called a translocation. Now in a translocation, we're going to switch genes. So again, we're twisting up, but we're twisting up with a different chromosome. So we're going to say that we switch genes with a non-homologous chromosome. All right, so you can have two different types of chromosomes. Okay, the first set that we're going to talk about is called an autosome. So an autosome is going to be any chromosome that does not relate to your sex. So it does not relate to the fact that you are male or female. Okay, in humans, wow, this is so very wrong. In humans, we have 44 total. Okay, we have 44 total, and that's pairs 1 through 22. Okay, and those, again, are going to code for anything that's not related to you being a dude or you being a girl. So anything like your hair color, your height, your weight, how big your feet are, eye color, anything like that. Now, sex chromosomes are going to relate directly to you being male or female. Okay, we're not talking about transgender, intersex. We're talking about what you're born with. Okay, so this is going to deal with things like body hair, how you go through puberty, what you make. Now, there is only one pair of these in a set of humans, and that is pair number 23. Okay, now you've got two options. If you're a girl, you're going to have two X chromosomes, and if you're a boy, you're going to have one X and one Y. Now, you're going to get the X from your mom and the Y from your dad. So every time we have a baby, there's a 50-50 shot that you get a girl, because you'll get an X from dad and an X from mom, or that you're a boy who got an X from mom and a Y from dad. All right, now, I know my note guide was short. You may have to carry on to a different page for this slide, but I can write small. So, but if you guys need to use the extra space, use the extra space. Okay, so a karyotype. This picture right here is called a karyotype. Basically, it's a picture of chromosomes. We use those to determine where genes are on an actual chromosome. So like, what is this green gene here? What is this red gene here? We also use it to show changes in chromosomes, like are there too many, are there too few? Now we'll spend a lot of time talking about karyotypes in the next few days. A couple of things that you need to keep track of. Okay, first off, we're going to talk about something called crossing over, which happens during prophase 1. And basically what's happening is that these chromosomes here, these pairs, are switching genes. So that's why there's like a little bit of red here and a little bit of green here, that they're not completely the same color. We'll talk more about it later, but just remember that they're switching genes happens during prophase 1. Now, sometimes those chromosomes don't separate the way that they're supposed to, and they create differences in a person's chromosomes. There are two examples: trisomy, which means that a person received 3 copies of a chromosome instead of 2, and then monosomy, which means they only received 1. So examples of those trisomies: Down syndrome is trisomy 21, Edwards trisomy 18, Patau's trisomy 13, Klinefelter's trisomy 23, meaning they got 3 copies in whatever number of chromosomes. Monosomy means that they got 1 copy of a chromosome. The only example is Turner's, and that's monosomy 23, meaning they only got 1 sex chromosome. This is an example of a human karyotype that needs to be glued into your journal. Double check your answers. But this will be on your quiz on Monday. Good luck. See you then.